We heard you are trying out for the baseball team today.
Yeah, I'm worried I'm not strong or fast enough though.
According to my research, protein shakes are proven to enhance muscle ability. We can help make you one.
But why are proteins so important?
Well, Ralphie, that's a good question. Proteins perform a variety of functions in our bodies. But I can show you better than I can explain it to you. Let's go on a field trip.
Oh no, not again.
Alligator nostril. Climb on the magic school bus.
Now we have shrunk down to fit inside the nucleus. This is where DNA is replicated.
What is DNA?
DNA is what dictates our genes. It is composed of nucleotides adenine, thymine, guanine, and cytosine.
Yeah, genes are why you have red hair, Arnold, and why Ralphie has brown hair. Also, we know because DNA is made of two complementary strands, adenine pairs with thymine, and guanine pairs with cytosine. It is just like a lock and key. I thought we were going to talk about proteins. How is this going to help me with my baseball tryouts?
Well, Ralphie, DNA is what codes for proteins. Therefore, DNA has to be replicated in order to make the proteins.
Yeah. But doesn't it start with transcription?
Right to the sea. Come on in and don't be shy.
First, you have to separate the strands of DNA.
That's right. Helicase is the enzyme that unzips the two strands just like a zipper. This is called initiation. Look, it's happening right now.
Wow. Now what is it doing?
Now, another enzyme called RNA polymerase III is adding the complementary bases to the template strand called the mRNA to make a new strand called tRNA. Every 3 bases on the mRNA is called a codon, which are like a genetic word. Anticodons are on the tRNA strand. Start codons are needed to start each strand transcription, and stop codons are needed to stop it. Look, now a 5' is added to the one end of the new RNA, and a poly A tail, which is made up of adenines, is added to the other end.
What does that do?
It protects them when they leave the nucleus, doesn't it?
It does. Also, RNA is modified by splicing if there are introns, which are parts of the DNA that don't code for proteins. Splicing cuts out these regions and it rejoins the exons. This strand does not contain any introns, though.
Well, why did it leave?
Well, now tRNA has to leave the nucleus to go to the ribosome. The ribosome is made up of 2 subunits, one large and one small. Let's go follow it.
According to my research, ribosomal RNA makes up 60% of the ribosome and is the most abundant type of RNA.
Indeed it is, Dorothy Ann. Translation begins when our tRNA strand binds to the P site of the ribosome. This initiates translation.
Look, it's happening now.
Yes, next it's going to go through elongation where the codons are read for amino acids. Then peptide bonds are formed between these amino acids. Then the polypeptide is transferred to the A site. It is held there until another amino acid comes along. Then it goes to the E site.
The E site, could that symbolize the end?
Indeed it does. Here water is added to release the amino acid. This occurs when a stop codon is reached. We call this termination.
That's it?
Yes, now it is the protein stop to travel to the Golgi apparatus. There it will be packaged and sent to where it needs to be used in the body.
Wow, I guess proteins really do make a difference. Let's go make that protein shake.
Yeah!
Step inside, it's a wilder ride.